Good morning to everyone. Today we are going to discuss about tRNA. Or this is another called adapter molecule. Adapter. First of all, say this tRNA. This tRNA actually we call it is adapter molecule or soluble RNA. Okay, this tRNA we call it is adapter molecule or soluble RNA, sRNA. Simply called we call here this is a soluble RNA. Soluble RNA. Why? Because means this tRNA read the genetic code. Read the genetic code. The nitrogen bases. We call codon. This codon we call it is the amino acid. It belongs to the amino acid. So that can be read by this tRNA. And also binds that means it's bind the tRNA main function one more. It binds the amino acid during the translation process. Binding of amino acid. That is also one of the function. Where it is the binding that you can see in the translation mechanism. First of all, the tRNA main function read the genetic code and it binding the amino acids. It binds to the amino acids. Next, the tRNA name, the adapter molecule is assigned to tRNA by Francis Crick. The scientist Francis Crick. Once again, see this point. The adapter molecule. Why it is adapter also I will show in the translation mechanism, next concept. The adapter molecule assigned to the tRNA by the scientist Francis Crick. Next, in the tRNA main, the bases. So many bases, particularly to determine the base sequence of tRNA molecule. To determine the base sequence of tRNA molecule. So, who the scientist name particularly is the Robert Coley. Robert Coley. Okay, so that determine the base sequence, particularly in the tRNA. It is done by the scientist Robert Coley. Next, many nitrogen bases. Okay, particularly many nitrogen bases means modified to form. In this tRNA structure, many nitrogen bases are modified to form. It is a coil-like structure. This coil-like structure you can see that we call it as a secondary structure. So, the secondary structure, how it is present means it is the clover leaf model. Clover leaf. Okay. So, many bases are modified to form a coil-like. You can see such a structure, we call it as a secondary structure. Such a secondary structure, how it is present means a clover leaf model. Such a clover leaf model, they are identified by the scientist Coley. Scientist who is a Coley. Next, one more point, basic point. Actually, this tRNA, it is an intermediate link where the tRNA is the intermediate link between the reading of codons on mRNA. Reading of codons on mRNA. This is the mRNA. Reading of codons on mRNA and the formation of amino acid chain for proteins. That is a polypeptide chain formatted. Here the tRNA is present. This tRNA some of the nitrogen basic remains. This tRNA it contains some bases. These bases are binds to on mRNA. So as a result, we have formed the polypeptide chain. So that's why here is the polypeptide chain proteins formation, and this tRNA binds to an mRNA. So that means in mRNA and here polypeptide chain formation. So here tRNA here act as a intermediate link. How it is that in translation mechanism I will show clearly. Okay, so this tRNA here act as a main intermediate link. How means here reading of codons on mRNA here, reading of codons on mRNA and the formation of amino acids for proteins. That means polypeptide chain formation and reading of codons on mRNA in between. So this tRNA present. So that's why this tRNA here it is intermediate link. It is the intermediate link. Okay, that's it. So this is some basic points for tRNA and how it is the diagram you can see. Actually this tRNA compact structure. Okay, the tRNA mainly the presence of it is a compact molecule. It is inverted L-like structure actually. Okay, it is an inverted L. So the tRNA is actually the bases are coiled. The bases are modified to formation of coil-like structure. That's how we call it as a clover-leaf model. Actually, the tRNA, these bases are it is a compactingly formed. It is an inverted L shape. Inverted L shape is present. So now this tRNA you can see structure. It is a the tRNA structure of this tRNA. It has 4 loops are present. How many? 4 loops. 4 loops are present. So first one it is a amino acid acceptor site. Amino acid acceptor. That is amino acid acceptor binding site. Okay, this is a D loop. This is what you call it as a D loop, and another one it is a T loop, and this is the anticodon loop. So here this is the amino acid acceptor site. That means amino acid binding site. And one more it is called it is the D loop. Second one, third one it is the T loop. And here fourth one it is the anticodon loop. So in this first one you can see amino acid binding site. So the amino acids are attached to the where it is a CCAN at a 3-OH group region. Okay, so in the 3- here the 3-OH group of CCA codon can be bind this site. This is called it is the amino acid binding site. Next D loop. This D loop we call it here. Okay, this D loop we call here it is a DHU. Dihydrouracil. What do you call here? Dihydrouracil. So it is a synthesizing site. D means it is a dihydrouracil site. Okay, that means this dihydrouracil, it contains D loop, it contains dihydrouracil, and it is a binding site for, this is a binding site for amino acyl synthase, it is the synthase enzyme, amino acyl synthase enzyme. Next, this one's loop. This loop is the ribosomes. It sits as the ribosome binding site. This is what you call, it is the ribosome binding site. Next, another one you can see. So this is the loop we call. It is the anticodal loop. So this is the anticodal loop means here, see here, the nitrogen bases are present. In this nitrogen bases, here the anticodal can be containing these are 3 nitrogen bases. These 3 nitrogen bases, it can be bind to the mRNA. That means see AUG. In this AUG, that means this is the anticodal loop. The anticodal loop, it can be containing the AUG nitrogen bases. So based on complementary, this is the mRNA. Based on complementary, this is the mRNA sequence. On the mRNA sequence region, so based on complementary on mRNA, so these nitrogen bases are bind to the mRNA. Then A means it is uracil. T means what do you call it here? A. Here G means C. So that means this anticodal loop, it contains nearly about, it is the 7 bases. Out of 7 base, 3 nucleotides. Particularly here, the loop, it is a binding site for it is a anticodal loop particularly. So this anticodal loop contains 7 bases, out of which 3 nucleotides forms anticodons. 3 nucleotides form anticodon, which will form base pair with complementary codon in mRNA during translation. So this is the 4 binding sites. So once again say, so this tRNA can be contained, it is the amino acid binding site, this is the T loop, and also this is the D loop, this is the T loop, this is the anticodal loop. So this anticodal, it can be containing that, what you call it is the AUC. Okay, anticodal containing AUC maybe. So based on complementary, so opposite sequence are present in mRNA. So this is bind to based on complementary. So finally it can be formation of what do you call it is the proteins. That means polypeptide chain finally it can be formed. So next, this is the structure of tRNA1. It is a 4 loops. In textbook it is not present clearly. I am saying it, what are the sites are present: amino acid binding site, D loop and T loop, anticodal loop. So that's it. Already previous what I said, that is the basic points of this is the tRNA. Okay, next we are going to discuss translation. What is meant by here the translation? So the process of polymerization of amino acids. The process of polymerization of amino acids to form polypeptide chain, that's we call it as a translation. Translation means the process of polymerization of amino acids to form polypeptide chain. That's we call it as translation. Very, very important. So translation means the process of polymerization of amino acids. That means the number of amino acids are formed. That's what I am saying. What do you call here? The process of polymerization of amino acids to form a polypeptide chain. That's we call it as a translation. So in this process, the sequence of amino acids, defined by the sequence of bases in the mRNA. Okay, so what is this? Here the sequence of amino acids defined The sequence of amino acid that is defined means operated by the base sequence in the mRNA. That means this is the mRNA. Okay, so here polypeptide chain is formed. So here polypeptide chain contains number of amino acids. So the sequence of amino acids that are defined, operated by, particularly where on mRNA, particularly on mRNA. Next, one more point. The polymerization of amino acids. The polymerization of amino acids. For formation, it requires some energy. The energy we call it is ATP. The energy we call here ATP. This energy where it is utilized. So here this is the amino acid. Amino acid between the bond is formed. So this is what you call it is a peptide bond. This is what you call peptide bond. The formation of peptide bond here require this energy. That means the amino acids are joined by a bond. This is what you call peptide bond. So for the formation of this peptide bond here require energy. That energy we call it is ATP. That energy we call ATP. Okay. Once again, see translation. The polymerization of amino acids to the formation of this polypeptide chain. This is the amino acid maybe. So the formation of the polymerization of amino acids to the formation of polypeptide chain. That process we call it is the translation. So the sequence of amino acids formatted. These are defined means operated by the sequence of nitrogen bases in the mRNA. So these amino acids joined by a bond, this is what you call it is a peptide bond. Okay, joined by a bond, what do you call? Peptide bond. So for the formation of this peptide bond, it requires energy. That energy we call it is ATP. Okay, so now here in the translation process, first of all I will draw the diagram. How it is polypeptide chain formation, we will discuss. This is the two DRNAs. This is the translational unit. This is the translational unit. This is the mRNA, you can see this is the 5' end, this is the 3' end. Okay. So here see in the translation unit, already I said what is meant by translation. So the amino acid CSC, the amino acid between the joined by a bond, this is called peptide bond. So the peptide bond for this formation it may require, it is energy that should be called ATP. So now in this translation process for the formation of polypeptide chain, the amino acids are activated. This one, see this is the amino, the tRNA binding this codon. The amino acids are activated in the presence of ATP, and also this codon, it can be linked to the cognate tRNA. So that's why we call here charging of tRNA or amino acyl tRNA. Okay, once again see this point. So in this, for the formation of this polypeptide chain, the first phase of its cell. So in that the codon, that means this amino acid, these 3 nitrogen bases we call codon. So these amino acids are activated in the presence of ATP. Okay, these amino acids are activated how? In the presence of ATP. So these amino acids are linked with the cognate means coordinated with tRNA. Such a bond we call it is a charging of tRNA or amino acid tRNA. Once again repeat see. So this codon is activated, that means this amino acid is activated in the presence of energy. It is also linked with cognate with tRNA. Cognate means coordinated with tRNA. This is such, that's why here we call it is a charging of tRNA or amino acid tRNA. Next, after that, in this here say 2 tRNAs are present. These 2 tRNAs are brought very close together for the formation of here polypeptide chains. Next, in the translation unit, cellular factory. What do you call here? Cellular factory. Cellular factory means this is the ribosome. What do you call here? Ribosome. This is the ribosome. See, this part is the ribosome. The cellular factory we call it as the ribosome. The ribosome main function it is synthesis of proteins. Actually, this ribosome consisting of structural RNAs. Okay, structural RNAs, 80 different proteins. Okay, so this ribosome containing of structural RNAs and 80 different proteins are present actually. Okay, so this ribosome consisting of actually is the structural RNAs and 80 different proteins are present. In its inactivin stage, that means these ribosome, these are inactivin stage. The ribosome, it can be consisting of, the ribosome it can be formed or consisting of 2 subunits. So this is the one subunit in the ribosome. This is the another subunit. That means this is a this is the smaller subunit, this is the larger subunit. Okay, so the ribosome actually contains structural RNAs and 80 different proteins are present. In its inactive stage, the ribosome it can be formed two subunits. So one is the smaller subunit, this one. Another one, this is the larger subunit. Another one, larger subunit. So this first one you can see, this is the mRNA sequence. This is the mRNA sequence. This smaller subunit encounters the mRNA. Encounters means encounters here, encounters an mRNA. Encounters means attaching an mRNA. Whenever the smaller subunit encounters an mRNA, encounters means attached here. So it can be starts the protein synthesis. That means it begins the protein synthesis. Next, this larger subunit. See this larger subunit. So this larger subunit having two sides are present. In the two sides, so it contains two sides. In the two sides, here see amino acids are present. Based on complementary, so appropriate, that means particularly it is the, what are the complementary base sequence upon, it is the nitrogen bases are, it can be added to the formation of polypeptide chain. Once again see, this larger subunit contain two sides. So these two sides are, it having, so the nitrogen base, the tRNA, two tRNAs are present, here are the nitrogen bases, that means these codons are bind to tRNA, so that means subsequently it can be added to the amino acids. The subsequently it can be added to the amino acids, it can be formed the polypeptide chain. Next, sometimes this ribosome directly it is act as a catalytic activity. That means this ribosome it can be act as a catalytic activity means here to the formation of it is a polypeptide chain. Polypeptide chain. Next, this is the mRNA. In the mRNA, this is the sequence of RNA. This is the sequence of RNA. This is, you can see, this is the mRNA. This is the sequence of RNA. That is RNA one of the type. It having the nitrogen bases sequence present. So in this mRNA having the starting codon, it is AUC. The starting codon, what do you call? Here AUC. Next, in the translation unit, 5' and before, that means here, and 3' and after, some additional sequences are present. Some additional sequences, that means nitrogen bases are present here. So the sequences are not translated. The sequences are, that is, the sequences are not translated. That means these sequences cannot perform in the translated process. That means cannot participate in the translated process. That's why we call it is untranslated regions. This is what you call untranslated regions. These three. That means these are. So in the translation unit, 5' and before and 3' and after, some sequences are present. That is nitrogen bases are present. These bases cannot participate in the translation. That is untranslated region. This is what you call untranslated. That's why cannot participate in the translation. So that's why such a region we call it is untranslated regions. Okay, why? Because these untranslated regions are present. So the translation process it can be going perfectly, accurately. Okay, the translation process it can be going accurately. That means the formation of polypeptide chain formation, accurately it can be formed. That's it. Okay. Next, in the how it is the this polypeptide chain form. See, actually, this mRNA, this is the tRNA, this is ribosome. The ribosome first bind with where it is the 5' end. That means in the starting region of mRNA. So the tRNA, okay, the tRNA, what are the sequences present? The sequences bind first of all here. So the starting initiator codon it can be recognized by this tRNA. Actually, how it is the actually requirements it is the AUC present starting here 5' end. Okay, the ribosome here again present this is the tRNA. This is another tRNA. So the tRNA what are the sequences for AUC? So we have what is the T already this amino acid is binding to tRNA. So what are the sequences present? This sequence is bind with the sequence. What are the this sequence is bind with the starting codon of AUC based on complementary and base pairing based upon. Okay, based on complementary based upon we are bind with the starting codon that can be initiated by which one? This tRNA. So after that what will happen whenever this one bind here? This is bind to another codon. So what will happen here? This codon is binds to here. This codon is binds to starting codon. This codon is binds to here another codon. What will happen? So what are the formation of this codon? It is can be comes to formation of amino acids to here. Between the bond, what do you call it is a peptide bond form. So this one is comes to here. So what is the AUC is comes to bind with the tRNA. Next, this one is another codon is bind with tRNA. That means this codon what will happen? This codon is comes to here. So between the formation of bond, peptide bond. Next, the ribosome move to codon here. Next here it can be bind. The tRNA here sequence it can be bind to here. This one is binds to next codon. So that is the ribosome move to codon to codon, codon to codon, codon to codon. So what are the amino acids? The tRNA bind with amino acid na appropriate codon can be bind based on complementary rule. Okay, based on complementary rule. So here form the polypeptide chain. So whenever this ribosome reaches to the terminal region, what will happen? Releasing factor. It is that whenever the ribosome reaches the terminator, that means where it is that stopping codon region, it can be reached. So the releasing factor can be bind. So this polypeptide chain is fall off. That's it. Okay. So how it is once again this one? This is very confusing part. So actually, so the tRNA. What are the amino acid is present? This amino acid is first bind to AUC region. That means based on complementary sequence only it can be bind. So what are the here sequence? Suppose it may be A is bind with A is U, T is bind with A. So this UAC. That means here UAC present. This UAC is present bind. This ribosome first bind with here. So this UAC bind with AUC. Next here CAA is present. CAA bind with which one? Here the GUU maybe. So here. So the CAA here bind with here. That means here tRNA and this is the tRNA. Here CAA is binded. So based on complementary it can be bind. So this UAA that means here comes to formation of amino acid between the peptide bond is formed. So this CAA can be comes to here. So here between the bond is formed. This AU is bonded to tRNA. GU bonded to tRNA. Again like this codon to codon, codon to codon, appropriate codon. What are the codons are present? Appropriate codon it can be bind based on complementary rule on the mRNA. So finally this ribosome is just to terminate region. So the releasing factor can be bind. So automatically what will happen? Stop the process of translation. So after that this polypeptide chain is separated. That's it. This is the process of translation. Once again say simply I will explain the process of polymerization of amino acids. Polymer, this is a polymer of amino acids. That's how we call it as a translation. So these amino acids that are defined, that is operated by the sequence are present on RNA. The formation of amino acids, it require energy. So the energy, okay, so the energy, that's we call it is ATP. And also the formation of amino acid between the joint by bond, this is what we call peptide bond. The peptide bond formation can be require energy, that's we call ATP. Okay, and next here the amino acid is linked with the tRNA. So this is what you call it is starting of tRNA or amino acid tRNA. Here amino acid synthesis enzyme is participating. Next, after that, so the ribosome, simply I am explaining, ribosome, the cellular factory. The cellular factory, what do you call it is ribosome. The ribosome main function consisting of proteins. So the ribosome can be consisting of structural RNAs, 80 different proteins. So these 80 different proteins inactive in space. The ribosome consisting of it is the two subunits. One is the smaller subunit, another one is larger subunit. So the smaller subunit is N counters and mRNA, which begins the protein synthesis, and larger subunit having the two sites are present. So in the two sites approximately appropriate, that means subsequent amino acids are bind to the formation of polypeptide chain. Sometimes the ribosome act as a catalyst. So that's why it is to the directly formation of polypeptide chain. So this is the sequence of mRNA. It is the sequence of mRNA. The sequence of mRNA is mainly flanked by the starting codon. Flanked means arranged by starting codon AUC. Okay, so here the mRNA 5' before, 3' and after some additional sequences are present. These additional sequences cannot translate. Such a region we call it is untranslated regions. So why? Because these untranslated regions are present means here particularly polypeptide chain formation accurately. So here in this how to formation polypeptide chain means so the tRNA what are the sequence amino acids bind. So that sequence is bind with the starting codon that can be initiated by tRNA. So and also these two tRNAs are bind with two codons. So subsequent codons are added to the tRNA. So finally it can be formation of polypeptide chain. So this ribosome codon to codon and appropriate codons are can be bind. So finally it reaches the it can be formed polypeptide chain. So finally it can reaches the terminal region. So releasing factor can be bind. So it can stop the translation process. This is the translation mechanism. Very important.